Dear students, in this video we are going to discuss about the structure and the function of hemoglobin. What is known as hemoglobin? Hemoglobin is nothing but heme protein. It is known as protein. So what is known as heme protein? Heme protein is nothing but the protein. Hemoglobin is a protein which contains heme group. That means heme group containing protein. Heme group containing protein. Okay, so we know about protein. What are proteins? Proteins are made up of amino acids, various amino acids, and all these amino acids combine together through peptide bonds. Therefore they can give proteins. But we have one doubt. So whether these proteins may be containing some non-protein parts, that means heme group. Yes, it is possible. Okay, therefore this heme group can be otherwise called non-protein part of non-protein part, or the other name is prosthetic group. It is known as prosthetic group. Okay, so this non-protein part or prosthetic group can be present in not only in hemoglobin, in any protein containing enzymes or some other important biological macromolecules. Okay, so let us look on to the structure of heme group. So what about the structure of heme group? Heme group, actually it is known as, heme group contains porphyrin ring. It is porphyrin ring as well as iron in Fe2 plus state. Now we have one more doubt. What is known as porphyrin ring? Porphyrin ring actually comes from porphyrin ring. Porphyrin ring. Porphyrin ring. Okay, what is this porphyrin ring? Actually, this porphyrin ring is nothing but it is actually a macromolecule ring. It is a macromolecule ring or macromolecular ligand. Okay, why it is known as a ligand? Because this porphyrin macromolecule ring is going to uh, coordinate with Fe2 plus. Yes, this Fe2 plus is going to be coordinated with the porphyrin ring. Okay, so what about the structure of the macromolecule porphyrin ring? It is actually, its structure is looking like this. Okay, this one is the porphyrin ring. This one is the porphyrin ring. This porphyrin ring actually contains 4 nitrogen atoms. Here there are 4 nitrogen atoms, and these 4 nitrogen atoms are coming and combine together to form this biggest macromolecular ring. And moreover, when these 4 nitrogen atoms are coming together, they will form this biggest macromolecular ring by forming 4 pyrrole rings. These are known as 4 pyrrole rings. Pyrrole rings actually looking like this. It is known as pyrrole. Okay, this is known as pyrrole structure. Okay, so in this manner, the porphyrin ring will be formed. Okay, then after that, let us look on to the structure of porphyrin ring. So this is the advanced structure of the porphyrin ring. This is known as porphyrin ring. Okay, there is no change between porphyrin and porphyrin rings, but a porphyrin ring contains the side chains. Here you can notice the side chains. Here you can notice the side chains here, side chains. Okay, so porphyrin ring gives the side chain derivative products is known as porphyrin ring. Okay, so what about the derivatives? We can notice here that COOH group and methyl groups and some ethylene groups. Okay, these are some side chains. And let us come to the, so this is known as porphyrin ring. So in the middle of this porphyrin ring, we can locate the Fe2 plus. All these four nitrogen atoms will make a coordination bond with this Fe2 plus. Okay, so this is known as one subunit. This is one heme group. So as I told you before, heme group is nothing but it is known as the porphyrin ring as well as iron in Fe2 plus oxidation state. Okay, this is one heme group. Okay, then what is the relation between this heme group and heme containing protein called hemoglobin? Actually, four hemoglobin containing four heme groups. Okay, four heme groups present in a protein. Okay, this four heme group containing protein is known as hemoglobin. Okay, so let us draw very simply this diagram. Fe2 plus is coordinated with the heme group, and this is known as one heme group. Okay, then the fifth coordination site, that means nitrogen 1, nitrogen 2, nitrogen 3 and nitrogen 4. Then fifth position will be coordinated from the histidine ring, from the histidine, not ring, histidine amino acid of the protein chain. Okay, this protein chain containing another histidine. Okay, this histidine will be again coordinated with one more heme group, and the same nitrogen from the another histidine group of the protein chain will be making one more coordination bond with another heme group. Then the same nitrogen will be again coordinated with one more heme group. So in this way we can explain the four subunits of hemoglobin. Okay, so let us conclude about the structure of hemoglobin. So the hemoglobin contains four subunits of heme group, with the protein chain. So far, in a particular or a single heme group, we have just discussed about the five coordination sites of the iron central metal atom. What about the sixth position? Okay, since this is an octahedral environment, therefore 1, 2, 3, 1, 2, 3, 4, 5. This histidine amino acid gives the fifth coordination site for the given iron central metal atom. Therefore, what about the sixth position? The sixth position is reserved for oxygen molecule. So during the respiration, whenever the hemoglobin molecule is coming in the bloodstream. When this bloodstream reaches the lungs, okay, the lungs containing blood and this blood containing hemoglobin will be automatically receiving the oxygen molecule during the respiration. Okay, therefore therefore one hemoglobin molecule therefore one hemoglobin molecule will absorb 4 oxygen molecules during the respiration. One hemoglobin. Why one hemoglobin molecule is receiving 4 oxygen molecules? It is because of one hemoglobin contains 4 heme subunits. Okay, so that's why it is receiving 4 oxygen molecules. Okay. Okay, so far we have just discussed about the structural importance of hemoglobin, and now let us extend our discussion to the structural change, right, during the entrance of oxygen molecule. Let us classify this structural change process as two steps. One is before the entering of oxygen molecule, and one more is after the entering of oxygen molecule. First, let us start with with our discussion with the absence of oxygen molecule. So, before the entering of oxygen molecule, the structure of hemoglobin will be like this. So all the nitrogen atoms are coming together to form porphyrin ring. So as soon as they form a porphyrin ring, they will be they are having a hole like this. It is a hole like this. In this hole, Fe2 plus must be properly fit exactly in this hole. Okay, so let us draw or let us consider Fe2 plus like this. Fe2 plus like this. Okay, so this Fe2 plus is somewhat very large, so large, and this cannot sit in this hole which is formed by 4 nitrogen atoms. So that what will happen? The 4 nitrogen atoms present in the hemoglobin molecule is just lying in the same plane. Okay, so can you imagine, these are these 4 nitrogen atoms are lying in the same plane. Okay, and the hole formed by these 4 nitrogen atoms cannot allow the bigger Fe2 plus atom inside of their hole. Therefore this Fe2 plus is lying just above the plane of these 4 nitrogen atoms. Okay, so this case can be discussed for absence of oxygen molecule. So before the entering of oxygen molecule, the biggest size of Fe2 plus cannot fit in the exact hole of the 4 nitrogen atoms present in the heme group. Okay, now let us extend our discussion why this Fe2 plus is so large that it cannot fit in the exact hole of the 4 nitrogen atoms. Why this Fe2 plus is so large? Why Fe2 plus is so large? Okay, so before the entrance, before the entering of oxygen molecule, Fe2 plus is under the weak field ligands, under the weak field ligands. So what will happen when the weak field ligands are approaching Fe2 plus? Their size will be usually large. Yes or no? Okay, it is because of the T2G and EG occupancy. Okay, so whenever weak field ligands are coming, then the given electrons will be jumping over to the EG orbital. Therefore, If all the electrons are accommodated in the T2G level itself, then the size will be reduced. But in the case of absence of oxygen molecule, yes, oxygen molecule is a strong field ligand. Because of the absence of oxygen strong field ligand, the atoms will be going to Eg level. Therefore the size is expected to be more. Yes, it is very large. So that's why Fe2 plus in the absence of oxygen molecule is very large, and that's why it cannot be sitting in the exact hole formed by 4 nitrogen atoms. Therefore this structural change has been existing. Okay, now what will happen when oxygen is entering in the heme group? So what will happen? Nitrogen, nitrogen, so they are forming a hole here, and whenever the oxygen is approaching the Fe2 plus, since we know that Fe2 plus is D6 configuration 1 2 3, and since we know that this delta O is large in the presence of oxygen molecule, the fourth electron cannot go to the Eg level, and this electron is again accommodated in the T2G level itself. That's why the Fe2 plus, the size of the Fe2 plus will be somewhat reduced. Since Fe2 plus size is reduced, it can easily fit in the uh, in the hole formed by 4 nitrogen atoms. Therefore, nitrogen, all 4 nitrogen atoms in the heme group, as well as iron atom Fe2 plus can sit in the same plane. Okay, now let us move on to our next discussion to the oxyhemoglobin and deoxyhemoglobin and their colors. Okay, so when when oxygen is incorporated with the heme group, then it is known as oxyhemoglobin. Whenever oxygen is not incorporated with the heme group, then that is known as deoxyhemoglobin. Usually oxyhemoglobin is red in color and deoxyhemoglobin is blue in color. Okay, then their magnetic properties. Whenever oxygen is bound with the Fe2 plus, Fe2 plus, it becomes oxyhemoglobin. Oxyhemoglobin is nothing but the Fe2 plus is surrounded by strong field oxygen ligand. Therefore, no unpaired electron is seen in either T2G or Eg. Therefore, it is diamagnetic in nature. It is diamagnetic in nature. But in the case of absence of Fe2 plus, absence of oxygen molecule, it is known as deoxyhemoglobin. So in the absence of oxygen, the extra electron can be accommodated in the T2G level, and also moreover there are 4 unpaired electrons. Since this deoxyhemoglobin system contains more unpaired number of electrons, it is paramagnetic in nature. So dear students, I hope you can follow this discussion on the structural importance of hemoglobin, as well as the structural changes, color, and the magnetic properties of hemoglobin. So dear students, let us meet in the next video. Thank you.